Imagine staring at a data set that, took a brilliant scientist 15 hours of just grueling manual coding to clean up.
Oh, it's painful. Just hours of staring at a terminal.
Right. Now imagine a system that does it perfectly in under 10 minutes while you're just drinking your morning coffee.
Welcome to the deep dive. Our mission today is to explore exactly how that time warp is possible.
Yeah, we're unpacking this really fascinating new paper from Nature about a groundbreaking AI framework called CellAtria.
And it really is groundbreaking. I mean, it's a profound shift in how we handle biological data. We're moving from a world where scientists basically have to be software engineers just to look at their own experiments to a world where they can just ask the right questions.
Exactly. But to really understand why this is such a massive deal for the listener, we have to talk about the data itself first.
Yeah, single-cell RNA sequencing, or scRNA-seq.
Right. Historically, looking at the RNA in a tissue sample was a bit like, drinking a fruit smoothie.
A smoothie. Yeah. Traditional bulk RNA sequencing takes a tissue sample, blends millions of cells together, and just gives you this average readout. So if you taste strawberry in the smoothie, you know there are strawberries in there somewhere.
But you don't know if there was like one giant mutant strawberry or 100 tiny normal ones.
Exactly. And you certainly don't know how that strawberry was interacting with the banana before you hit blend. You lose all that crucial context to the individual components.
But then single-cell RNA sequencing comes along, and suddenly we aren't drinking the smoothie anymore. We have a pair of magical tweezers, and we're examining every individual piece of fruit, cell by cell, looking at its unique genetic profile.
And the leap in resolution there is staggering. We can suddenly see cellular heterogeneity. We can spot the minute differences between individual cells across healthy tissues, or, map out the kinetic microenvironment of a cancer tumor.
Or track exactly how specific cells respond to experimental drugs. It's incredible.
It is. And because this technology is so powerful, public databases are just overflowing with this high-resolution data. Mountains of it.
And here is the massive bottleneck. While the data is sitting right there, actually using it is a nightmare.
Oh, an absolute nightmare.
To reuse a published data set, you have to manually read the paper, hunt down the metadata like the tissue type or the species, figure out the specific database ID, download these huge files, and then write custom scripts just to get the data into a usable format.
Which requires a very rare dual skill set. You need the biological domain knowledge to understand the complex paper, obviously, but you also need serious computational proficiency to write the bioinformatics code.
Which means the work just gets handed off to specialist bioinformaticians, creating massive delays. But I know what you might be thinking listening to this. It's the 2020s. We have artificial intelligence.
Right, the ChatGPT question.
Yeah. Why can't a scientist just copy and paste the paper into a large language model and just ask it to write the analysis code? Hasn't AI already solved coding?
Well, that is the exact trap a lot of research labs are falling into right now. Relying on an LLM to dynamically generate ad hoc analytical code on the fly is, it's highly dangerous for science.
Break that down for me. Why is it so dangerous?
Beyond the well-known issue of hallucinations, where the AI just confidently invents a function that doesn't actually exist, you have to deal with temporal cutoffs.
Meaning the AI's training data is out of date.
Yeah. If you've ever spent a week trying to get an old piece of software to run on a new operating system, you know the frustration. Bioinformatics software evolves incredibly fast. A package that worked perfectly in 2023 might have entirely different syntax today.
So if the LLM was trained on the old syntax, the code it generates will just break.
Exactly. But the real deal breaker is reproducibility. If you ask an LLM the same question twice, you might get two slightly different scripts.
Oh, and in a clinical setting, that's a huge red flag.
You simply cannot base a cancer drug target on code that changes every time you prompt it. It completely violates strict quality control guidelines.
Okay, so letting the AI write raw code is scientifically risky. That brings us to how the researchers behind CellAtria, a team at AstraZeneca, solved this. They completely changed the AI's job description.
They did. They used what's called an LLM-mediated tool-centric paradigm. It's a fundamental architectural shift. Instead of treating the AI as a programmer, they treat it as an orchestrator.
Here's how I picture it. Instead of the AI being a lone-line cook frantically trying to chop vegetables, read a recipe, and manage the stove all at once, and, occasionally mistaking salt for sugar, CellAtria treats the AI as an executive chef.
That's a perfect analogy. The executive chef doesn't do the chopping. The AI takes the order from the customer, the scientist, in plain English.
But to execute the order, it delegates to a team of highly trained, strictly regulated sous chefs.
Right, and in CellAtria, these sous chefs are a library of pre-vetted, robust analytical tools.
The paper mentions this is built on a LangGraph-based multi-actor architecture. Now, for those of us who aren't computer scientists, what does LangGraph actually do here?
Think of LangGraph like an intricate flowchart for a factory assembly line. The AI doesn't just do tasks randomly. It follows a strict one-way path where step A must be completed and verified before it's allowed to hand the data off to step B.
So it's locked into a safe sequence.
Exactly. The language model handles the natural language from the user and interprets the intent, but it never writes the raw math. It invokes specific pre-written schema-validated tools to do the heavy lifting.
Okay, let's do a quick recap so far so we're all on the same page. Number one, single-cell RNA sequencing data is amazing, but it's incredibly hard to wrangle manually. Number two, letting AI write raw analytical code is scientifically risky because of hallucinations and reproducibility issues.
Right.
And number three, CellAtria solves this by using the AI not as a coder, but as a smart dispatcher, an executive chef for strict, safe tools. So moving on, how does this executive chef actually read a menu? How does it get the data?
Well, the paper tests 3 specific scenarios for this document-to-analysis flow. Let's say you have a web link to a longitudinal study looking at the immune responses of 2-month-old infants following routine vaccinations.
Okay, scenario one, a web URL.
You just drop that link straight into the CellAtria chat. The system autonomously goes to the web page, retrieves the HTML, and uses tools to filter out all the digital noise, the CSS, the random website scripts. It isolates the actual text of the study.
And then it just reads it.
Yeah, the LLM reads it to extract structured metadata, tissue type, species, disease state, and the crucial database session IDs.
And it does this conversationally, right? Like it anticipates what you need and suggests actions.
Exactly. It might say, I found the GEO database ID for this infant study. Should I download it? And then it pulls the data directly from the public repositories.
But as anyone who has tried to do research knows, half of the internet is locked behind academic paywalls or messy university logins. You can't always just scrape a web page.
Which brings us to scenario 2, local PDFs. The user just uploads the PDF directly into the interface. The researchers tested this with a really dense study on T-cell states in lung cancer patients.
Oh, so it bypasses the internet entirely.
Yes. CellAtria uses a built-in document parser called PyMapPDF. It physically reads the text layer of the offline PDF, completely ignoring web rendering issues and publisher paywalls.
That is such a huge frustration saver.
And in this specific test, instead of just grabbing the whole massive study, it extracted fine-grained sample-level data called GSM data, using specific identifiers to pull only the exact data sets the user wanted.
And then there's scenario 3, right? Direct integration. Say I'm browsing a massive pre-curated collection like the CZ Cell by GeneDiscover platform, and I already have the download URL.
In that case, it skips the reading phase entirely. It goes straight to file integration, grabbing the files and organizing the directories.
Okay, I have to challenge this a bit. Scientific papers are notoriously messy. They're unstructured, sometimes terribly written, and often missing key information. What happens if a paper just totally forgets to mention the specific tissue type? Does the AI just guess?
That is where those architectural guardrails we talked about really shine.
Yeah.
The system has a built-in safeguard to prevent speculative inference. Guessing leads to hallucinations, and hallucinations ruin data pipelines.
So what does it do?
If fields are missing or hidden in unstructured jargon, CellAtria defaults to marking them as unavailable.
It just admits it doesn't know.
Exactly. It forces the human to fill in the blank, rather than quietly making a bad assumption.
You know, I don't know is sometimes the most scientific answer an AI can give. So once CellAtria has perfectly extracted, downloaded, and organized all this raw data, where does it go?
It gets handed off to its partner. This brings us to the engine room of the whole operation, CellExpress.
CellExpress, the pipeline that actually crunches the numbers.
Right. It transforms these raw, massive matrices of genetic counts into biological insights using an industry-standard workflow.
Let's walk the listener through exactly how CellExpress does this, because it's not just magic math. The first step is quality control, or QC. I was actually surprised to learn that not all cells survive the sequencing process intact.
Oh yeah, sequencing is a harsh physical process. A lot of cells burst or die. So CellExpress filters out the junk. For example, it automatically removes cells with high mitochondrial gene content.
Why mitochondria specifically?
When a cell is highly stressed or dying, its membrane breaks down and it essentially dumps mitochondrial RNA into the sample. If you don't filter those out, your final data will be completely skewed by dying cells rather than healthy biological signals.
Makes sense. It even hunts down things called doublets using a specific tool called scrublet.
Yes, scrublet is brilliant. A doublet is exactly what it sounds like. Two separate cells that get physically stuck together in the sequencing droplet. To the machine, it looks like one giant weird hybrid cell with twice the genetic material.
How do you even find that in a data set of millions?
Scrublet works by mathematically simulating artificial doublets from your clean data and then hunting through the real data to find and remove anything that matches that artificial profile.
Wow. All right, so the dying cells are gone, the weird hybrid cells are gone, the data is clean, but we are still dealing with thousands of genes per cell. Human brains cannot visualize a 20,000 dimensional graph. How does CellExpress shrink that down?
It uses dimensionality reduction. The specific tools here are UMA and TSNE.
I've always thought of them like cartographers creating a flat map of a highly complex 3D globe.
That's a great way to visualize it. They mathematically squish the high-dimensional data into a 2D scatter plot, ensuring that cells with similar genetic profiles are clustered together in the same neighborhood.
And then there is the problem of batch effects, which I found fascinating. If you sequence half your samples on a Tuesday and the other half on a Thursday, sometimes the data looks wildly different just because of minor changes in room temperature or, like, a different batch of chemical reagents.
Right. Those artificial differences can ruin an experiment. So CellExpress uses a tool called Harmony to smooth them out. Harmony essentially looks at the different batches, finds their mathematical centers, and aligns them so that a T-cell sequenced on Tuesday overlaps perfectly with a T-cell sequenced on Thursday.
It aligns the math so the biology can actually speak.
Exactly. And the final step is looking at those neat little clusters and automatically labeling them. It uses tissue-agnostic models like similarity to say, based on the gene expression, this cluster right here is composed of T cells.
And it uses tissue-specific ones too, right? Like cell typus.
Yep, for when you need a highly specific annotation based on the exact organ you're looking at.
Now, hearing all these complex automated steps, a skeptic out there is likely saying, this sounds like a giant black box. Data goes in, magic math happens, and a result pops out.
And they'd be right to worry about that.
I completely agree with the skeptic. I wouldn't trust an AI driving my car if I couldn't see the steering wheel or the road.
The creators of CellAtria anticipated that exact fear. To prevent the black box problem, they built a highly transparent, human-in-the-loop user interface. It's divided into 4 critical panels, so you can see exactly what the machine is doing at all times.
Let's break those down. The first panel is the chatbot window. That's your plain English conversation with the executive chef.
Right. The second is the real-time log viewer. This is a running ticker showing you exactly what tool the AI is invoking at any given second and whether it succeeded or failed.
And the third?
The third is the agent backend panel. This is like reading the AI's mind. It literally shows you the internal reasoning step by step and how it translated your English request into those specific tool commands.
But the fourth panel is the one that really blew my mind. It is an embedded terminal and interactive file browser. You are chatting with an AI, but right there on the screen is a live command line shell.
It is the steering wheel. If the AI names a file something strange, or if you want to manually tweak a parameter, you can jump directly into the shell environment, navigate the folders, and debug the code without ever leaving the app. You have your hands on the controls the entire time.
Okay, time for our second quick recap. 1. CellAtria safely extracts data from messy papers. 2. The CellExpress engine perfectly cleans, clusters, and annotates that data using tools like Scrublet and Harmony. 3. A transparent 4-panel dashboard ensures the scientist never loses control.
Spot on.
So that all sounds amazing in theory, but what happens when you actually put a stopwatch to it? Let's talk about the scale test.
Well, the benchmark run is genuinely hard to believe until you see the data. They went back to that infant vaccine study we mentioned earlier. They gave CellAtria the URL and essentially just said, go.
A completely autonomous run, no human intervention at all.
None. CellAtria read the paper, found the database ID, downloaded the datasets, set all the context-aware parameters for the CellExpress pipeline, and executed the entire analysis.
From raw URL to fully annotated, quality-controlled, visualized single-cell data.
Yes, and it took less than 10 minutes.
Wow.
The researchers benchmarked this against a human bioinformatician doing the exact same workflow, manually reading the paper, downloading the files, converting formats, and writing the scripts. The human time was roughly 15 cumulative hours.
15 hours down to 10 minutes. Just think about the implications of that for a moment. If you are a cancer researcher, getting 15 hours of your life back for every single data set you want to look at isn't just a time saver. It fundamentally shifts what you spend your day doing.
You stop fighting with data formats and start actually doing biology.
Exactly. And they proved this wasn't just a one-off fluke, right?
Right. They stress-tested the system at a massive scale. They unleashed CellAtria across 25 public data sets spanning 6 major cancer types: breast, lung, prostate, colorectal, ovary, and pancreatic.
That's a ton of data.
In total, it processed 290 individual samples, resulting in about 1 million post-quality control cells.
I want to dig into how they measured its performance during that massive scale test, because it wasn't just about speed. They measured the AI's consistency using a metric called the Gini coefficient.
Yes, the Gini coefficient.
Now, usually you hear about Gini coefficients in economics to measure wealth inequality, where a score of 0 means perfect equality.
Exactly. But in this paper, they adapted it to measure the variance in the AI's outputs, specifically the number of tokens it generated and the file sizes of its responses across all those different data sets.
Why does token variance matter?
Because if the AI was getting confused by the complex data, it might output 500 words of panicked guessing for one data set and only 10 words for another. The Gini coefficient tracks that variance. A high score means the AI is unstable and hallucinating.
But the results showed a Gini coefficient of around 0.10.
Which is incredibly low. A score of 0.10 means the AI is acting like a highly tuned machine. It uses almost the exact same computing effort every single time. It didn't wildly fluctuate in verbosity.
It behaved systematically and predictably across 25 completely different, highly complex cancer datasets.
Yep, averaging about 1.45 minutes per reasoning task using GPT-4o.
It proves this isn't just a fragile prototype, it's an industrial strength framework. So what does this all mean when we step back and look at the bigger picture? By strictly separating the language model's conversational skills from the rigorous, pre-vetted math of the CellExpress pipeline, CellAtria completely democratizes bioinformatics.
It really does.
Yeah.
It removes the middleman between the scientist and the insight. It shifts the burden of data ingestion from a small pool of specialist coders directly to the bench scientists.
The people who actually understand the biology the best, but maybe don't know how to write a complex Python script.
Exactly.
Which leads me to a really provocative thought, and something I want you to mull over after this deep dive is done. If agentic frameworks like CellAtria can perfectly handle the how of complex data processing without us writing a single line of code, how is this going to change the way we train future scientists?
It is the million-dollar question for universities right now.
Will coding skills eventually become entirely obsolete in biological research? Will the value of a scientist shift entirely away from syntax and scripting, replaced sheerly by the ability to ask an AI the most profound, creative biological questions? We are finally putting down the blender, and instead of fighting to see the fruit, we just get to ask what it tastes like. Thank you so much for joining us on this deep dive. Keep questioning the data, and we'll see you next time.